Hello, welcome. We'll just finish today the part belonging to the enzymes which can be motivated by the presence of the vanadium. So, how vanadium can get inside the cell, how vanadium can show some interactions with different important biological processes. So, basically today we will be talking about the interaction of all these groups related to the vanadium. So, the part 4 of vanadium enzymes, and if we consider some of these as the model compounds, because the particular active site what we can have in the biological system. So, if we have the active site in the biological system and some of these positions having some useful donor atoms like nitrogen, like oxygen or any other useful group which can be identified by different techniques like X-ray structure or other spectroscopic signatures. So, how a particular active site can go for the vanadium entrapment and how the vanadium can go for several important reactions which can also be mimicked in the laboratory by synthesizing some related model compounds. So, the synthesis and characterization of these model compounds plays some important and interesting role in understanding several important aspects related to vanadium chemistry, that if we just simply consider whether this active site is participating or catalyzing some important reaction where electron transfer plays some important and major role, then we should have a related model compound where the environment, the coordination environment compared to the active site environment, where definitely these groups are the immediate donor atoms which are bound to the metal center, the metal ion center. But in case of model compound, these are the useful donor atoms, can be nitrogen, can be oxygen, and how these particular donor atoms can be a part of the ligand system. It can be a part of the ligand system, and when the metal center is not present, that means several other oxygen or nitrogen donor atoms may be available there, may not be originating from the same ligand system, that means the same protein backbone, but some other small groups can enter over there where the same passage of the metal ion, that means the passage of metal ion as vanadium, can also be studied very nicely. So, these model compounds play some vital and important role in understanding the type of chemical reaction which is being catalyzed by these vanadium centers for electron transfer or some atom transfer reactions. So, if it is an atom transfer, so we can also see the same atom transfer reaction with the help of these model compounds. So, one such example for that is that when these model systems can have some water as the ligand, and if water is occupying a position which is correspondingly trans to the oxo group, which is important for the labilization from the strong trans influence. So, we have chosen one such system which is reported in the literature also, that if we have this backbone as a salicylaldehyde backbone with bromine substitution to the para position of the phenol oxygen, and which is being condensed with 1 mole of glycine. So, we get very simple way ONO tridentate ligand. So, this is one part of the cavity what we are just talking about, that this particular part is available for coordination of the vanadium. So, if the system, the cell can permeate the entry of vanadium center to this particular part, so vanadium can go and immediately form 3 bonds. One is this vanadium oxygen, second is this vanadium nitrogen, and third is this vanadium oxygen bond. So, the same is also true when vanadium is bound to this particular ligand system in a typical coordination compound. So, what happens there, that immediately depending upon the nature of the vanadium salt what we are using over there, immediately this vanadium is coordinated to this tridentate ligand. So, the binding of this tridentate ligand is very important. So, model compounds we can have from the choice of these ligands. So, first of this is a simple tridentate ligand. So, why we are taking this particular tridentate ligand is that we have this oxygen nitrogen oxygen binding, and vanadium is forming 3 bonds to that, and we do not know what other positions would be and what are these positions will come and bind to the vanadium center. So, this particular phase, if it is tridentate to vanadium and 2 more positions are occupied by 2 other groups which are unknown, which can be X or which can be Y. So, this particular species, that means X and Y, we already know all these things, that X can be our very good substrate species. So, when this ligand bound system, that means the vanadium is there and ligand was already present there. So, depending upon that, this VL species can have 2 to 3 positions vacant. It is not vacant in the true sense, it can be occupied by water molecules which is present in the system and which is already there when vanadium is bound to these water molecules to give us the corresponding ACO compound. So, these vacant sites can go for binding this X and some other reagent, this Y can be our reagent. So, there are positions where the substance can bind to directly to the vanadium center, reagent can also bind to the vanadium center, and in the next step these two, that means the reagent and substrate can react to give rise to our product. So, product is formed from there. So, very important thing is that how this tridentate ligand is binding to the vanadium center. So, we all know that depending upon the flexibility of the ligand backbone, we can have 2 different types of binding. One is meridional binding, that means these 3 can bind to the vanadium and they are in the same plane. So, when they are not in the same plane, the same tridentate ligand occupy one particular phase. So, if they occupy one particular phase, we call this as a corresponding facial binding. So, the choice of ligand is therefore can dictate us whether we should take a corresponding tridentate ligand which can bind in meridional fashion, or the same tridentate ligand which can go and bind the metal center in the facial mode. So, in this particular case, what we see that this particular ligand as well as the other ligand where we have some different part of the glycine derivative, we have some alcohol part also, to check whether this vanadium has higher affinity for this carboxylate oxygen. This is the carboxylate oxygen, or it has some binding affinity to the alcohol oxygen. So, when we get the compound, when we synthesize the compound and characterize the compound, we can identify that vanadium has more affinity for the carboxylate oxygen rather than the alcohol oxygen, because these two have different pK values and the basicity of the remaining oxygen what is getting from there as O minus are also different. So, vanadium will have direct affinity for binding to the carboxylate oxygen. So, when these three positions are blocked in a meridional mode, we can have three other remaining positions, and these three remaining positions are interestingly suited over there, that if we get the immediate vanadial oxygen, because the vanadium compounds very easily form the corresponding vanadial complex, it is directly coming from the oxygen of the water molecule also. So, initially if we have the vanadium water bond, then after deprotonation it goes to vanadium hydroxide and then finally to vanadium oxo bond. So, when oxo is there, and if we just see that three of these are coming from the ligand, fourth is also another aqua molecule. So, initially when this dotted water molecule is not present, we have a situation where the geometry of the coordination environment can be considered as a square pyramidal one. So, if this particular vanadium oxygen bond is not so short, and if we cannot have a corresponding pyramidal distortion of the geometry, this water molecule can come opposite to that of our vanadium center with respect to the oxo group and weakly start interacting with the water molecule. Similar thing also happens with this compound number 5, where this vanadium, instead of water coordination, it can bind to some alkoxide anion. So, this is anionic coordination, and also during hexa coordination it has some weak coordination from the water molecule trans to that of our vanadium oxygen double bond. So, this particular occupancy, that means why this water molecule should be there or not, that is being dictated by the oxo group. So, this particular presence of this oxo group, if it is not transforming from oxo to hydroxido or hydroxide to aqua molecule, this oxo function can weaken the corresponding vanadium oxygen bond coming from the water molecule trans to this vanadium oxygen double bond. So, what we get that we can make these compounds in the vanadium plus 5 oxidation state. So, initially what we get, if your ligand is tridentate dinegative, in this form, this ligand, this L, if we can have charge on this oxygen and charge on this oxygen, we can have a corresponding compound of the ligand where L is giving rise to 2 negative charges. So, vanadium is there. So, we can have 2 negative charges from the ligand, and also since it is coordinating with the oxo. So, vanadium oxo can also 2 negative charges. So, immediate neutralization of 4 negative charges by vanadium 4 plus, and if it goes for one more step towards oxidation, it can go to vanadium in 5 plus oxidation state. So, modeling of this particular oxidation state is very easy, and we can have the corresponding vanadium pentavalent compound, that means vanadium in plus 5 oxidation state, if we can have an O4 donut set in a roughly trigonal bipyramidal coordination geometry. So, what should be the most preferred coordination geometry? So, instead of this particular square pyramidal or octahedral geometry, the N4O donor set, here also we can have 3 oxygen and 2 5 oxygen and N. So, this is NO5 donor set, this is also NO5 donor set for an octahedral geometry around this vanadium as well as on this vanadium. But if we can have a NO4 donor set around the same vanadium center, it can distort itself to a corresponding trigonal bipyramidal geometry, which is a more preferred geometry for this particular vanadium center. And we also see we can compare these vanadate groups or the vanadate anions to that of our phosphate. And the phosphate anions we will see in case of phosphate ester hydrolytic reactions that this vanadium center will have some preference for trigonal bipyramidal geometry compared to square pyramidal geometries. So, here this particular levelization, that means the presence of vanadium oxygen bond, how they are weakening the corresponding vanadium water group can be seen if we determine the corresponding single crystal X-ray structure of these synthesized compounds in a good crystalline form. And determination of this vanadium oxygen bond, this particular bond, this dotted bond, we find that the corresponding bond distance for this vanadium oxygen bond lies in the range of 2.28 to 2.35 angstrom, which is clearly a bigger value, a longer value, where we see that the normal distance falls in the range of 1.98, because within the same system we can have another bond which is this particular vanadium oxygen bond, which is cis to this vanadium oxygen double bond, but this bond is pretty shorter in the range of 1.98 angstrom compared to this bond which is in the range of 2.35 or 2.28. So, this particular bond is pretty weak, and when substrate comes and bind to this particular center, it immediately can displace these two positions. That means either these two water molecules can be replaced, one can be considered as the binding of X, the substrate, and another can be bound as the corresponding reagent for the usual transformation centered at the vanadium site. So, interestingly also that this particular vanadium complexes, if they go for some amount of interaction, what we see that already we have this vanadium oxygen double bond, and we can have other groups. So, if the plane, the square plane is formed from 3 other donor atoms, one is nitrogen and 2 oxygen, and this other oxygen. So, if 5 positions are occupied, and if one of these positions can be taken up by the corresponding substrate molecule, and we see that this particular center having some vacant site, that means this particular area or this particular space is very important, such that we can consider a corresponding binding of peroxide anion. So, we have seen large number of vanadium compounds, whether it is in the living organism or in some model compounds, that vanadium is forming a corresponding peroxide bond in this particular fashion, that means in the cis mode, and it is forming a very tight bonding pattern for a 3-membered ring, because this is a corresponding 3-membered ring for the corresponding coordination of both the oxygen atom, instead of any such bridging type of coordination involving 2 vanadium centers. So, we can have this, and this particular group, basically this peroxide compound, which has a corresponding charge of 2 minus. So, the vanadium peroxo compound is very important, in this case, and we should have sufficient vacant space such that the peroxide anion can go and bind to vanadium in this form. So, once it is bound, we have this oxygen and this oxygen. So, we need further stabilization. So, if possible, that means this particular linkage is not very much stable compared to our oxo binding, oxygen binding or nitrogen binding. So, to stabilize this particular peroxo group to the vanadium side, we need some other type of interactions which can stabilize the entire vanadium peroxo fragment. So, what is that? We see that in this particular case, this vanadium peroxo group, and we already know that it has 2 negative charge. So, some residual delta negative charges are there which is present on these oxygen atoms, and if we have some group attached to this pyridine ring of the ligand system, that means if we have some ortho NH2 function. So, these NH2 groups, if they are available, so when it forms the corresponding complex, this NH group is in close vicinity to this particular oxygen and it can form some hydrogen bonding interaction. So, through such hydrogen bonding interactions, this particular vanadium peroxo species can be stabilized, and that also gave us some important information that if this particular group is our reagent and if this particular vanadium peroxo reagent can show some hydrogen bonding interaction on the substrate molecule, then this particular substrate can be reactive towards this activated peroxide anion which is bound to the vanadium group. Similarly, not only the NH2 function of the ligand system, but simple water molecules. So, if we have water molecules as well as protons present in the systems like that of our 3-dimensional structure of the ice molecule. So, one water molecule over here which is already hydrogen bonded to another molecule of water, and we can have the corresponding proton, either this is bound to this carboxy function. So, if the carboxy function which is already bound to the vanadium center have instead of O minus group, it can have the OH function, and this OH function can be stabilized by this fast water group of this water dimer. So, this is the water dimer which is getting stabilized through hydrogen bonding, through internal hydrogen bonding between two water molecules, and this oxygen further shows hydrogen bonding interaction with the OH group of the carboxy end, and the peroxide linkage showing some other type of hydrogen bonding interaction which is different from compound 9. That means, this hydrogen is forming interaction with both the two oxygen atoms of the peroxide group. So, it is a different type of stabilization of the vanadium peroxo unit. So, not only this stabilization helps this particular entity, but it also helps the presence of the OH function or the carboxy end of the ligand, because in the long polypeptide chain or the protein chain, we can have several of such OH groups attached to the polypeptide backbone. It can be originating from the amide backbone, or it can be originating from the corresponding carboxy backbone. So, these backbone hydrogen atoms available on oxygen or nitrogen, if it is amide, can further be stabilized in the presence of water molecules, and these water molecules can further interact with the peroxide anion to stabilize this vanadium with respect to that of our peroxide link. So, we call these interactions, these hydrogen bonding interactions, we all know that these interactions we call as non-covalent interactions. So, non-covalent interactions are beyond the molecular interaction. So, we call them as the corresponding supramolecular interactions. So, we have the vanadium compound is one molecule, and then we can have some other part of the species like this NH2 group or the water molecules coming from the separate water molecules, it can be present in the system as the lattice water molecule or some other water molecules which is trapped within the crystal lattice for that. So, this interaction with the ligand periphery, that means the peroxide functions are present at the ligand periphery because this particular part is blocked. Only the part which is available for these hydrogen bonding interactions is that side where the peroxide linkage is coordinating to vanadium. As already I mentioned that if we can have some vacant space or vacant area in this particular area, then only the peroxide can show this sort of coordination to the vanadium center, and which further shows hydrogen bonding interactions with the water molecules. So, hydrogen bonds in all these complexes play some important role in stabilizing the entity where we can have the direct coordinate bond between vanadium and the peroxide anion. So, these peroxide groups we can have. So, in this particular entity where we can have the corresponding vanadium as the corresponding vanadium oxo function and the peroxo function. So, we can consider this as vanadium oxo peroxo species. So, if we can go for some reaction where we want to study the corresponding oxo transfer reaction, we can see that this particular oxo group can be transferred to some other substrate. So, substrate should be there and we can transfer the oxo group to the substrate. In some cases, if we have the species which is a composite of both the oxo group as well as the peroxo group, we can see that whether this particular oxygen atom is being transferred from vanadium center from this oxo end or from the peroxo end. So, several of these reactions, this peroxidase activity we have seen, which has been obtained in case of vanadium chloroperoxidase, vanadium bromoperoxidase species, where the vanadium peroxo group is interacting with the corresponding Cl minus and Br minus, which is found from the marine origin. So, if we consider the same oxo transfer reaction to bromide, that means the bromoperoxidase activity, or if we just consider the corresponding oxo transfer reaction on the sulphide group, we can see that they are all catalyzed by the oxo vanadium species through some hydroperoxo intermediate. That means, initially if we can have a non-oxo vanadium compound, that non-oxo vanadium compound can be transformed to some oxo vanadium compound, and that oxo vanadium compound can interact with hydroperoxides, forming the corresponding stabilization of the peroxide unit, either the neat peroxide anion or the protonation of one end giving the hydroperoxide unit, which is originally started giving us through the hydrogen bonding interaction of the peroxide anion to some hydrogen bond donor. So, what we see that this is the particular corresponding transition state where now the vanadium is present in a trigonal bipyramidal geometry, not in some square pyramidal geometry or octahedral geometry, but it is present in a typical trigonal bipyramidal geometry. So, all these species are very important. That means, if we can have this nitrogen, we can have these as one donor atom and another is to this side. But also if we have the simple nitrogen atom from the protein chain, and this particular position can be occupied by simple hydroxide group, this is oxo group, this is the second hydroxide group and this is O, this is the third hydroxide of function, and it can go for deprotonation giving O minus. So, if they react with hydrogen peroxide, what happens that when hydrogen peroxide is bound to the vanadium center, this position of this nitrogen getting changed, it is coming from this particular position, is occupying this particular OH group, and nitrogen is over here, this oxygen O minus is moving over here, and one new water molecule is coming over there, and this has been converted to a corresponding vanadium oxo function, and which is trans to this particular water molecule that we have seen just now, that the labilization of this water molecule in a typical octahedral environment is due to the presence of this vanadium oxygen double bond or the vanadium oxo form. So, from a trigonal bipyramidal geometry, we move to a pseudo octahedral geometry, because this particular bond is little bit squeezed one, and this we can also consider as a one bond. So, if we consider this as one bond, so it is the trigonal plane, and these are the two sides for the trigonal bipyramidal geometry. Otherwise, we can consider, if these two are considered as two bonds, this can be considered as a typical example of octahedral geometry. So, when we put some proton, that means this particular peroxide anion is getting protonated, and the center what is generated over there is our vanadium oxo plus hydroperoxide anion. So, this is not the simple peroxide anion, this is now the hydroperoxide anion. So, this hydroperoxide anion is useful to react. So, this is the most reactive species, and this reactive species is basically responsible for interacting with the substrate group. If the substrate is the bromide anion, which can supply this particular HO groups, is the giving you HOBr species. So, OH group is coming from here, and this peroxide group due to this particular reaction is going back to this oxo hydroxide species. So, peroxide species is therefore no longer will be present over there. So, this peroxide group is getting consumed over there, and we have the corresponding transformation of Br minus to HOBr. Similarly, this oxo can transfer to the sulfide group R2S. So, sulfide group is also taking this O plus species, that means this particular part of the peroxide hydroperoxide unit, giving rise to this immediate attack on the sulfur, and finally to the sulfoxide formation. So, that we know that how we can transfer this oxygen atom to simple dimethyl sulfide group to form the corresponding dimethyl sulfoxides. So, DMSO reductase reactions, we already know that DMSO reductase type of reaction where the sulfides are getting transformed to sulfoxides can also be seen through this sort of reaction, where vanadium is interacting through a vanadium oxo peroxide unit. Therefore, the presence of all these three groups, that means the presence of this nitrogen, presence of this oxo group, and the presence of these peroxide linkages are important for the reaction where we transfer this HO plus unit to Br minus or SR2 unit. So, if we just consider what is this nitrogen, if this nitrogen we can consider that the corresponding imine nitrogen of the ligand system in the model compound, or this nitrogen can be considered as the corresponding enzyme nitrogen of the histidine and the nitrogen function of the model compound. So, the presence of this nitrogen is crucial, and which is important, because in all these cases whatever species we are getting in the form of the oxo hydroxo species, which is reacting with the substrate molecule, is all these cases not only the vanadium oxo peroxo unit is present, which is all the time bound to the nitrogen, and this nitrogen is coming from the enzyme histidine, or it is coming from the imine group of the nitrogen ligand. So, all these vanadium oxo peroxo species, we have some unique nitrogen donor atom. So, this is therefore crucial for all these compounds. We can have the specific nitrogen donor which can control the reactivity, and this nitrogen can come from the enzyme also, and this can also come from the model compound. So, what we see next, that some of these vanadium compounds can show some insulin like effects. So, the condition where insulin is responsible for glucose metabolism is very important, and this particular biomolecule can tackle the corresponding degradation of glucose molecule in the case of glucose metabolism reaction. So, the effect of this insulin can be mimicked if we consider some vanadium compound, some important vanadium compound can be a good substitute of this particular insulin compounds by showing some insulin type of effects. So, what we know that in this particular case, the action of insulin, which is responsible for our carbohydrate and lipid metabolism in our body, when insulin is inactive in our body, is not interacting, we can have some diabetic condition, that means the insufficient carbohydrate metabolism and lipid metabolism. So, action of this insulin molecule is important, and how it is acting on this carbohydrate or lipid molecule is that during this interaction a series of signaling processes are taking place. That means, when insulin is entering into the system, it can give rise to some signaling process, and that signaling process is responsible for the assimilation of the glucose or the carbohydrate molecule to the system. So, basically this particular insulin is interacting with the insulin receptor system. So, there are some receptor sites, and those receptor sites are useful to bind this insulin molecule, and this insulin goes and interact with these insulin receptor site. So, if we can have some equivalent vanadium compound of that nature which can function as an insulin, so these vanadium compounds can have some property of interacting to these receptor sites. So, that we see that some of the vanadium compounds which are of very special type, and we can choose these vanadium compounds from a large number of such vanadium compounds varying from one particular ligand system to the other. In this case, these vanadium compounds show some insulin like effect, and therefore can show some enhancement of the effect of insulin by two-step things. One is the phosphorylation of the insulin receptor, because how this particular molecule, that means the vanadium compound, which is a synthetic compound, coordination compound of vanadium, can go and interact with the insulin receptor through the corresponding phosphorylation step, and in some case it can inhibit the protein phosphatases. That means the protein phosphatases are responsible for phosphate hydrolysis reaction, and if we are able to inhibit or stop the corresponding hydrolytic reaction of these phosphatases, we can consider that these vanadium compounds are useful in inhibiting the phosphatase activities on the phosphate ester groups. So, therefore, this action of insulin is directly related to the corresponding interaction with the insulin receptor, and vanadium molecules with some interacting sites, which we have seen that it can interact with some site where it can show some hydrogen bonding interaction. In a similar fashion, these synthetic compounds can show some interaction with that of our receptor site. So, we can see that very simple compound, that means the vanadil sulfate, not a complex metal complex. So, vanadium sulfate is the corresponding typical inorganic compound where we have the vanadium oxygen group. So, we have the vanadilyl function. So, vanadilyl function is present, and therefore, in this particular case this VO is present as 2 plus, and in this particular case also what we have seen in the compound where we have the corresponding ligand environment, this was our ligand. So, we have the ligand environment, and in this ligand environment we have the same vanadyl unit is present. So, this is a bigger molecule or rather complex molecule, and if we consider that this should go and pass the cell membrane to enter into the cell to show some interaction which is similar to that of our insulin molecule. Before that, if we just consider that the simple vanadyl compound where the vanadyl sulfate, so we can check whether the vanadyl sulfate with VO2 plus unit can interact to the corresponding receptor site for the insulin molecule. So, when we use vanadyl sulfate, we see that the simple vanadyl sulfate can show some glucose control activity with some people, or it is on the experimental animals like rat, it can experimented on some rat to see that the type 2 diabetes can be controlled by the simple interaction with the vanadyl sulfate molecules. So, that gives rise to the typical medicinal applications of these vanadium compounds. So, in vitro and in vivo studies both inside the cell and outside the cell, we can study the corresponding activity for glucose control of these vanadium compounds of two different types of patients. One is type 1 diabetic patients and another is type 2 diabetic patients. In the former case, they have the corresponding insulin deficiency. That means their pancreas cannot produce sufficient amount of insulin to control the glucose metabolism, and in the second case it has some insulin tolerance. So, both these two types can be seen whether they can show some effect if we use vanadyl sulfate as a medium for controlling the glucose. So, they can stimulate the glucose intake into the cell. So, how we can go and we can put the glucose inside the cell. So, glucose should enter into the cell, and then we can go for the degradation of the glucose, and thus it can lower the corresponding blood glucose level of the diabetic patients. So, these diabetic patients have high blood glucose level, and this high blood glucose level can be counteracted if we can take the glucose inside the cell and we can go for the metabolism, and in this process it can thus interact with the glycogenesis and glycogenolysis. So, in these two cases, which is also related to lipogenesis, where the lipases are hydrolyzed. So, all these hydrolysis reactions, that means the glucose hydrolysis reaction and the lipid hydrolysis reactions can be controlled by the simple administration of vanadyl sulfate. So, some of these sites can be activated, and through that activation we can see that it can show some corresponding mimetic reactions. So, insulin, the molecule which can produce in Langerhans islet of pancreas. So, there is some site where pancreas is responsible for the production of insulin, and basically this is a peptide hormone, and it has a typical organic molecule only having a peptide backbone only, and which regulates the glucose and fatty acid or the lipid metabolism. So, insulin is responsible for both the glucose and fatty acid metabolism, and if we can have shortage of insulin, we cannot see the corresponding control of this glucose metabolism. So, this is a typical flow chart for several of these complex reactions, and where we see that the signaling cascades, we have considered that during this particular interaction it shows some signaling pathway. So, signaling cascades are important, and where we have the phosphate ester bond which is responsible for showing that signaling process. So, in the first step in figure A, that means this part of the figure which shows that this is our insulin molecule. So, this hormone is coming and within the cell membrane it is coming, and this is our receptor site. So, this particular part is the extracellular part and this is the intracellular part, and this particular part is responsible to put this particular glucose molecule inside the cell. So, when this insulin is coming and sitting comfortably on the receptor site, and receptor site has some tyrosine part which is very important, and this tyrosine part which is basically responsible for showing this particular receptor site whether this phenol group, this is the OH function, whether this OH function is phosphorylated or not, that basically gives us some information whether this insulin can come over here, and whether this particular gate, this is the gate for the entry of the glucose molecule from the extracellular site to the intracellular site. So, in this particular case, this PTP is nothing but protein tyrosine phosphatase. So, PTP, that is the protein tyrosine phosphatase, is the corresponding enzyme showing the corresponding phosphatase activity. That means, if we have the oxygen phosphorus bond of the phosphate ester, the PTP can interact with this and can break this oxygen phosphorus bond, showing the phosphohydrolysis reaction. So, this phosphoesterase reactivity can be seen if we can break this particular oxygen phosphorus bond. So, during that process, when we have this oxygen phosphorus bond, it shows the corresponding signal pathway, and this signaling pathway can open up this particular gate, and this through this particular path we basically, this arrow shows which is nothing but the transport or reaction path. So, basically this path, when the gate is open, insulin is sitting over here. So, when the gate is open, the glucose molecule can come and enter through this gate. So, here was the glucose here in the figure A. So, in figure B, we have the glucose molecule which is entered in the intracellular region, and this particular one molecule, when it is entered within the cell system, can go for the corresponding burning process. That means, the glucose is metabolized. So, glucose metabolism can only take place where we can have this particular glucose molecule inside this particular cell. So, this PTP, basically this PTP, then if it can work on this oxygen phosphorus bond, can responsible for this corresponding metabolism. So, insulin basically giving us some information that when insulin sits over there, the gate opens up and glucose can enter inside the cell. So, in the figure C, where we see that the insulin is not there, the gate is closed, and the PTP, if the PTP is available, the PTP can work on this and which can cleave the corresponding ester bond. So, the PTP is active, and the activity of the PTP is responsible for the breaking of this oxygen phosphorus bond, giving rise to the corresponding tyrosine unit. Tyrosine, that means the bearing the phenol group, and the phosphate anion. So, this situation we do not like. So, if this situation is there, we require the insulin to come over here to open up the gate. So, if PTP is active, it should break the corresponding bond, and if this bond is no longer there, the signaling cascade is stopped. So, what happens that if we have some vanadium compound, so if the vanadium compound is there and which can pass through the membrane, and this vanadium can go inside with some modification, which is curly bracketed vanadium and which is square bracketed vanadium. So, some modification on the vanadium compound can take place through its passage through the cell membrane, and this vanadium can interact with the PTP. So, the enzyme PTP is now interacting with the vanadium, and this vanadium compound can be considered as the corresponding phosphatase inhibitor. So, this phosphatase inhibitor, that means these PTP molecules will no longer be utilized for breaking this OP bond of the phosphate ester. So, OP bond will remain intact, and it shows the corresponding signaling process, that means it can produce the giving the signal to open up the corresponding gate. So, in absence of insulin, only the presence of this vanadium, which is inhibiting the corresponding activity of the PTP. So, the gate is opened up and the glucose molecule can enter inside the cell and can go for the corresponding metabolic pathway. That means, the glucose molecule can be metabolized, producing carbon dioxide and water molecule. So, these vanadium compounds therefore showing the same effect what we have seen in case of the presence of the insulin molecule. Only thing that it basically inhibiting the corresponding action of these PTP molecules. So, this PTP, if we can go for the corresponding inhibitor design for that, so we engage this PTP with this vanadium, so it is no longer available for the hydrolysis reaction. So, insulin mimetic behavior we can see from these vanadium compounds, and the entire scheme basically what we have discussed just now can represent a simplified, it's very simple form of signal pathway which is induced either by the insulin or by the vanadium. So, this path, this dotted line, the signal pathways can be seen only when we have insulin in our system, or we can have the corresponding vanadium compound within the system. So, if we have the cell membrane which contains the transmembrane receptor for insulin, and which is basically the corresponding tyrosine kinase. So, this tyrosine kinase is responsible for insulin docking, and insulin is docking is nothing but insulin is coming and sitting at the receptor site of the insulin, and tyrosine is getting basically phosphorylated, and during this phosphorylation it gives rise to a complicated signal transduction cascade. So, the dotted line what we are showing in the previous scheme is that it is basically a corresponding signal transduction cascade pathway, and this phosphorylation can be counteracted if the PTP, which is responsible for the hydrolytic rupture of the phosphate ester bond, that means the phosphate ester hydrolysis reaction, and during this phosphate ester hydrolysis reaction the signal process is taking place. So, this particular hydrolysis reaction is fully effective in absence of insulin or in case of insulin tolerance. So, how we can control this hydrolytic reaction? So, we bring the vanadate. So, the vanadate can come and enter the cell via some phosphate and sulfate channels. So, these are not some receptor sites. Unlike the insulin molecule, insulin needs some receptor site. Through that receptor site, insulin can enter the cell, but in case of vanadium, these are the phosphate and sulfate channels, which are very important channel sites, and through those phosphate and sulfate channels, vanadate can enter into the system, and through that entrance, the vanadate inhibits the phosphatases, and one such mechanism is that inhibition of PTP by vanadate only, and which allows the phosphate ester bond and thus the signal transduction to remain intact. The signal transduction should remain operative, which allows the gate to open and which can bring the corresponding glucose molecule within the cell for glucose metabolism. So, that is the basic function for all these vanadium compounds for glucose control, and which is showing some corresponding insulin mimetic activity. Thank you very much.